so many people to come to the public lecture. Yeah, I think that's going to be a very exciting evening. Yeah, with a lot of thought-provoking lectures. Yeah, but we're going to get that in a moment. Yeah. So what we thought. Yeah. We have this big virus evolution and molecular epidemiology workshop. Yeah. And we decided that our keynote speakers, that would be great if they could be open to the whole university. And we can really see that work. I can see potentially like almost like close to 100 extra people here. So I'm just going to give a 5 minutes introduction about the workshop per se. Yeah. So I think that's polite that I introduce myself. Yeah. My name is Túlio de Oliveira. I am a professor here at Stellenbosch University and the director of the Center for Epidemic Research Innovation, which is the organization hosting the workshop this year. Yeah. But if you go to this workshop, which is called the International Bioinformatics, Virus Evolution and Molecular Epidemiology Workshop, a mouthful, so we decided just to make called VEME, yeah, it's a very long-lasting program, yeah. It is a program that has been hosted in 18 different countries, in 27 different editions, yeah. So this workshop, it moved around the world, yeah. It started around 27 years ago by Professor Annemiek van Dam at University KU Leuven. Yeah, Professor van Dam is there taking a lovely picture of that. So she started the workshop as a one-day program, yeah, and then go to 2 days, 3 days, 4 days, and then the 7 first editions were in Belgium. And then the workshop started growing and moving around the world, yeah. So it has been hosted twice in South Africa. It also has been hosted in Brazil, Trinidad Tobago, Panama City, and many, many countries in the world, yeah. So, and the workshop grow, yeah. So now we have 4 different modules. It's hands-on computer analysis or discussion that involve multiple lectures, and people come from all over the world, but I'm gonna give you the statistics, yeah. So as the workshop grow, yeah, and Professor Annemiek van Dam also asked for other co-organizers to help to take over. So the last 3 editions, yeah, the last 2 of the editions have been organized by Professor Luis Carlos Alcântara, which is here. Yeah, Luis, don't know if you want to send to him. So not only he organized the last 2 editions, both in Rio de Janeiro, Brazil, that hosts twice, but in Panama City, in Panama. And he helped me to co-organize this workshop in South Africa. And last year, we are very excited to say that's going to be moving to the University of Washington in Seattle, going back to the US. And then what we would like to do, I don't know if you can see the map, is start filling some of the blank maps, especially in Africa or Asia or Australia, yeah. And one thing that we are excited, I don't know if Javier from Instituto Pasteur is here, that one of the things that we are discussing is to take to the Pasteur Institute, potentially to Senegal. So we have to be very nice to him, yeah. So this workshop, it's almost like, as Cheryl Baxter, the head of my scientific program, described, almost like a one-to-one tutorial, yeah. We have 51 lectures and organizers, yeah. And they come from everywhere in the world. So this year we have them represented for 13 countries, yeah. And they come, most of them have been teaching here for decades, yeah. And they're really passionate about trying to give back to the scientific community, yeah. And then this year we had 103 participants, yeah. From, sorry, it's the wrong letter there, that come from 16 African countries, 6 Latin African countries, 3 Asian countries. In total, we have 35 countries represented in this workshop. But what makes very excited for us is that by hosting Africa, and especially with the support of some of the funders, such as the Rockefeller Foundation, the World Health Organization, Abbott, and SLA, we got 43% of the participants are from Africa. And with support of the Pan American Health Organization, 20% of the participants in Latin America. And why do we need they get trained? Because that is a lot of the place that the next epidemics will emerge, yeah? And then what we do, this program that we are hosting here today, so we also provide 58 scholarships, so flying 58 people from around the world to come here, full scholarship or partial scholarship, yeah. And this is part of a much bigger program that we have been running in the last few years. So one program that we have been running, it is a very large capacity building program. It's too large to have one funder, so we have a basket of funders, which we are very happy with the participants, the African participants on this program. We have brought and trained 403 fellows from 48 African countries. And they come for training such as sometime weeks, workshops, such as this one, or middle-term fellowships that they stay 1 month to 3 months, or 1 year to 3 years, that's the long-term fellowships. Yeah? And we are really excited that this program is really happen to really build capacity in Africa, yeah, and now also in Latin America. And what the students come, a lot of time, if it's dry work or if it's mobile work, they will come here to Stellenbosch University. Otherwise, they will go to another absolute phenomenal building that have been completed at the medical campus that hosts CERI, which is the large genomics facility in Africa. So that's the way that we can give back not only data analysis, that's what we are learning here, but also the production and analysis of the data. So that's just a small introduction to the workshop per se and our capacity building program, but really you are not here to listen me. You are here to listen to the world experts, yeah. And the first lecture, it's going to be presented by Professor Edward Holmes, yeah. For the ones that work in our field, or the ones the kind of mad ones that don't believe in origin of virus, Ed Holmes are very famous, yeah. So just to give idea, he did his all undergraduate and PhDs in the United Kingdom. He did his first degree at University of London with a PhD in Cambridge. And then he moved as a professor to University of Oxford. And I was very lucky that he was my promoter when I took a fellowship at Oxford University in 2004. And I was so excited to work with him. And then I arrived there, and then he told me that he was leaving. Yeah, I hope it was not for that, yeah. But it was still a great fellowship. Yeah, the funny thing is that at least he gave me his office, which you have been for many years. So every day someone come and look. Oh, Ed is growing some hair. Yeah, so it was very exciting. Yeah. But then after that, he moved to the US, and then he moved, and he's now established at University of Sydney, where he's a full professor. He's also a fellow of the Australian Academy of Science and the UK Royal Society. Yeah? And Professor Edward Holmes has multiple awards, including the New South Wales Premier Prize of Science and the Australian Prime Minister Prize for Science, among others. So he's going to be our first keynote speaker. And then after that, we're going to take questions from her. And then we're going to have another professor that is as good as Eddie, yeah, but with a much better hair. That is that's Professor Marion Koopmans. Yeah, Professor Marion Koopmans is also extremely famous across the world. Yeah, she moved into virus, but her first degree was in veterinary medicine at the Utrecht University, where she stayed all the way to her PhD. Then after that, she became a fellow of the US Center for Disease and Control. And then she moved back to the Netherlands to be the chief of virology of the National Institute for Public Health and Environment. And then also a professor at Erasmus Medical Center. Yeah. Koopman was appointed by the WHO to go in the investigation of the origins of COVID. She stayed, I think, locked in quarantine for 15 days in China before she could go in and investigate that. Yeah. And she has really work in the interface between human and animals. Yeah, she's a member of the Royal Netherlands Academy of Arts and Science and the Royal Society of Science, and she has many awards, including the Machiavelli Prize and the Iris Medal for Science and Communication. So she's going to be our second speaker, and she will follow from Professor Ed Holmes. But now, before I tell any more jokes, let's invite Professor Ed Holmes.
Thank you for that abusive introduction. So what I want to do today, I'm going to do two things. What I wanted to talk about initially was just the diversity of the viral universe that I call the virosphere. But Tullio insisted that, and I always do what Tullio says, I also talk a bit about COVID-19 origins, which I can never, ever escape from. So I'm going to do that. So the first half of my talk will be about general viral biodiversity stuff, and the second half we focus on the origin of SARS-CoV-2. Now, in fact, you're lucky to get me because the last public lecture I was invited to give was in US Congress in July this year, and they kindly invited me to fly to Washington for Australia at my own expense, and to sit there for 4 hours and be harassed. I declined that kind of invitation, but they very nicely put my kind of name tag there just to kind of make sure that you can see that I wasn't actually there. So you're lucky to get me this time. So as I was saying, the first part's about just understanding the virosphere in general. Some of these slides will be a bit technical. I apologize if it's a bit too dense. I'll try and do my best to kind of make it quite general. And the second part will be directly on this. Okay. So what I do a lot of, and the reason why I'm here in the course, it's an amazing course, by the way, and Tullio deserves and his team an amazing amount of credit for what they do. It's quite extraordinary. What I try and do is use genomics to understand the diversity of this virus universe. And there are viruses all around us, as you'll see shortly. And what I'm particularly interested in is not COVID-19 or human diseases, but viruses that infect other animals. So if you take a tree of the metazoa, tree of animals that's up there, the vast majority of the viruses that we know about are from arthropods, like mosquitoes and ticks, because they cause diseases, and also chordates, right? Because like fish, I'm a chordate, you're chordates, I hope you're chordates. We eat chordates, they're our pets. So, and there's lots of other phyla, other branches in that tree that are just never, all these things down here, worms, I'm not really good at zoology, sponges, mollusks, they're just never studied, okay? So our view of viruses is very, very biased towards particular taxa. Now, in the last few years, what's happened is there's been a technique that's called metagenomics. I won't discuss it today. It's simply you sequence all the genetic material in a sample. And that's revolutionizing how we discover viruses. And it was that technique, in fact, was used to discover SARS-CoV-2. I'll discuss that shortly. And this graph here is just showing you the number of virus sequences that have increased dramatically in time over the last 20 years. And these little boxes here are some of the technological innovations that have happened, different types of sequencing. But you can see a massive increase in genome sequence data. But still, our understanding of viruses outside of things like us and things that bite us is actually very, very limited. So what I normally do for a living is try and redress that balance by asking kind of three questions. How big, how large and diverse is this virus sphere? What determines the diversity you see, and how do viruses move through whole ecosystems, I'll show you later on. And also, how do viruses occasionally jump host species and emerge to cause disease like SARS-CoV-2? So that's what I'm going to do today, right? And the way I often do this, as do a number of people now, is take the natural world and I sample particularly places where humans and animals, particularly wildlife, interact. So this is, you can imagine the kind of, sorry, it's not very clear, but you can imagine the kind of a landscape here. You have humans, you have wildlife, and the arrows are where they interact. And I like to think of those arrows as being like kind of fault lines, like earthquakes, like in earthquake science, it's points of stress and tension. And that's where the interactions are. That's where viruses can move between them, very occasionally cause disease. So I tend to sample there. And we sample, then we do sequencing. I won't discuss that today. Here's an example. This is my colleague Zhongzheng Zhang, who was one of the first people to sequence SARS-CoV-2. And here we've just gone to a town in southeastern China. And we sampled lots of rodents in that town. And then if you sequence them, this is a very, very difficult to see evolutionary tree of coronaviruses, including, of course, SARS-CoV-2. And you can't see it, but there's some ones in blue, and some ones in red, and some ones in green. And they're all from rodents. And the key thing is they're all from one city, one quite small city in China called Longquan, are literally around people's houses. And they're constantly jumping between species. So as we live as humans, literally under our feet are animals, and those animals carry viruses, including coronaviruses, in the case of these rodents from China. So that kind of shows you the diversity of the virus world right beneath us. And my kind of goal in science is trying to understand that, how viruses move between those species. Okay, so that's kind of what I do. Now, I'll just give you, the first part I'm going to do is just talk about the overall diversity of the virus field. I'm going to focus mainly on RNA viruses like SARS-CoV-2 or flu or measles, because they're kind of cool, I have to say. Now, the first thing that's to understand is first question is how large is the virus? How many viruses are there? How diverse are they? And the answer to that is bloody enormous, okay? And the way you can do that, this is the current, this kind of weird thing, is a current phylogeny of RNA viruses that they use to classify viruses, because viruses are classified like everything else. Now you don't need to know any of these kind of words, these made-up words. They're the kind of taxonomic groups. And at the moment we have 5 phyla of RNA viruses, like we have 5 of animals, and there are 5 more proposed. Okay. Now those of you that know your sequence, know your evolution, this tree is based on the most conserved gene in RNA viruses, called the RNA-dependent RNA polymerase. And even though that's the most conserved gene, these sequences are amazingly divergent. The average pairwise distance is often less than 5%, okay? And sometimes as low as 1%. Now the random expectation of if you align any 2 amino acid sequences is 5%. So these sequences are no more similar often than random. Okay, so if you're doing a tree, you might as well ask a random passerby in the street what the phylogeny is, because you'll get the same accuracy, because it's that divergent. So what I want to know is are there viruses that are so divergent you can't actually see them because they're just so divergent in sequence? Okay. So imagine here's my tree again, and that kind of outer edge around that with the orange circle there, that's the edge of where you can detect using a sequence of virus. Beyond that, out here, they're so divergent you can't see them because the sequences have evolved so far away. So are there viruses out there that are so divergent you can't normally see them? And the way we're trying to do that, trying to detect them, is using protein structure, because proteins are more conserved than nucleotide sequences, and protein structures are more conserved than primary sequences. So the idea is maybe protein structures are more conserved than the sequences that make this tree. So maybe we can find that. See that blue circle? Maybe that blue circle is the hidden variation, the hidden virus sphere that we can detect if we look for conserved structures rather than sequences, right? So that's kind of what we try and do. And this next slide is a bit technical, and so I'll do it very briefly, in part because I don't actually understand how it works. But what we did here, this is my colleague Mang Shi, and what Mang did with the help of the Alibaba Corporation, who gave an enormous amount of computing resources to this, is devise a new way of detecting highly divergent viruses in sequence data, viruses you can't see normally. But so what we did was actually, with Alibaba, was devise, everyone uses AI. My students email me via AI nowadays. It's extraordinary, right? ChatGPT. So what we used was an artificial intelligence approach to try and find these divergent viruses. What you do is you take a protein structure and you train the method to recognize these viral proteins. Okay, you train it using machine learning, artificial intelligence, and then you throw those trained programs at sequence data, see if you can find highly divergent viruses. And in doing that, we found 180,000 new viruses. Okay, 180,000, yeah. Right? Yeah, it's a very large number. Now, Now, some of those data were published already, and some we sequenced them ourselves. And we made sure that they were only in the RNA form, not DNA forms. We actually think that RNA viruses, including multiple new. So I remember there are 5 phyla of RNA viruses. We found, at least sort of 10 more. Sorry, we found about 180 phyla, including 10 of RNA bacteriophage. There are 2 at the moment. So an enormous expansion in the viral universe. So to kind of make it look a bit nicer. These kind of blobs are the kind of viruses we found. And you can see the kind of grey, the kind of grey inner, it's like those colorblind charts, right? I'm colorblind. The kind of grey inner circle, you see that there? That is basically that tree there. That's the extent of that diversity, right? And then using these new artificial intelligence methods, we can kind of go out further. So the orange, that kind of outer edge there, that is the edge of the virus universe that we can detect, right? As 100, so it's much bigger. All this stuff here is blue and orange. They're completely new taxa we can discover using this approach. So there are way more viruses out there you can ever imagine. Another way you can do it is by using evolutionary trees. Sorry, it's a bit complicated again. Each of these, you should basically, that tree I've shown you there, you shouldn't really do that. It's so divergent, you can't really align these sequences. So what you should do is lots of kind of mini trees rather than join them up. So here are lots of mini trees. All we need to see is the ones are in green are known, and the ones in yellow are new. And you can see there are whole tracts of the universe that you've never seen before that are in yellow. So we're finding lots and lots of massive new diversity doing this, right? And this won't be the end, there'll be more than this, right? Okay, so that's all a bit esoteric. Now I'm trying to make it a bit, kind of bring it home to you a bit. I'm going to focus on one group, if you can see there. And you can see I think you can read it says Nido or Nido. And these are the nidoviruses. Now you've probably never heard of them, but they're the viruses that have screwed your life up for the last 3 years, right? And because they contain the coronaviruses like SARS-CoV-2. So this is that tree I've just shown in the previous slide. I've kind of made it blown up, making it a bit easy to see. You've got some ones in black branches, ones in red branches. The ones in black branches, the ones that were known before our study. The ones in red are new, okay? So here are the coronaviruses up there. That one up there, that SARS-CoV-2, that kind of pathetically uninteresting little branch there. That's the one that's put us in trouble for 3.5 years. SARS-1 is next to it. There are some other families here, the ones in black. One I'll discuss in a second are the Arterioviridae. But you can see there's lots of this kind of hidden diversity. What you can then do, I guess this bit is for the virologists in the audience, what you can then do is then take that tree and try to use it to understand some other bits of biology about these nidoviruses. And in doing that, you find out that a few things. First of all, we normally think of these as being animal, particularly mammalian. But in fact, most of the viruses, nidoviruses, are actually found in sediments or aquatic samples. So they're not mammalian at all. We have no idea where they come from. Second, they're very, very long. These, we have one actually here is at 47,000 nucleotides, which is one of the longest RNA viruses ever found. There's one even bigger one, someone's found at 54,000. So they're found in very strange environments, and they're very long. They also have very complicated genome structures, more than they ever thought before. So now I'm going to blow up on these ones up here, even more kind of interesting. So here are the coronavirus. Oh, it's actually, I'm just going to skip that slide and move on a bit because I'm going to be too slow otherwise. So ignore that. Oh, by the way, I do a lot of sampling in many countries, including Australia. And we've been recently sampling this. Can you see the color of that? That's pink. That's Lake Tyrrell in Victoria. And it's a hypersaline lake dominated by archaebacteria. And we're sampling that lake to try and find RNA viruses that might infect archaebacteria. So it's actually dominated by archaebacteria. Anyway, I'll skip the rest of this, so don't worry. So now I'm going to blow up a bit more on the coronavirus part of that bigger tree. And again, we normally think of coronaviruses, because we just had one as being mammalian, but I've not got a glass, but in fact, excuse me while I take this drink. Coronaviruses are actually found in many different animals. So here, there's SARS-1 and there's SARS-2 there. All I've done in this tree, I've color-coded the backbone of the tree by the vertebrate group that they're from. So these ones are here, like SARS-1, SARS-2, you see. They're in pink branches, they're mammalian. Then next to them, we've got some ones in, whatever colour is green. They're from birds and reptiles. But down here, which people didn't realize until recently, there's a whole other set of coronaviruses in the kind of blue branch, and they're basically from wet things, okay, like fish. And most of all, which I really like, there's one there, it's called Canna coronavirus, and that's from a lamprey. And a lamprey is a jawless vertebrate. And that kind of phylogeny suggests that these things are probably as old as the vertebrates. Okay, so you think of these viruses being very recent, but they're actually ancient. These could be at least 500 million years old. So that's the coronavirus. On that tree I showed you before, I mentioned a thing called the arterivirus, is the next virus group down. You've probably never ever heard of arteriviruses, but in fact they're actually very, very serious, including where I live in Australia, and because they cause a very nasty disease of possums. I'm going to play a little video now if you watch this. Do you see that? So that's a brush-tailed possum that has an encephalitis, and that's caused by an arterivirus. Okay, these are very common backyard animals in Australia. And they will die. It's a very serious disease, and that's caused by an arterivirus that's kind of in this nidovirus group. And what's so interesting about these is if you then do a phylogeny of the nidoviruses, this is one of the other groups I've blown up again. What's very interesting is the phylogeny of the nidoviruses, the arteriviruses, completely matches the phylogeny of the hosts. Okay, so you've got mammals there, marsupials there, you've got fish and what's that? I can't really see what that is, reptiles there. So that again suggests these are absolutely ancient. Okay, and in fact, if you actually go back to that tree I showed a second ago of the nidoviruses, I think these kind of branches here are probably more than 500 million years old. So these viruses that you think are recent are in fact absolutely ancient things. Viruses have been with our planet, since life was first here. And that's true of actually other systems. So that's the nidoviruses, like coronaviruses. That antiquity, that ancient history is true of other groups. Another very famous virus that kind of periodically screws us up is influenza. Now, influenza is a virus, that's now classified in an order called I hate these names, the Articuvirales. That's over there, okay. And people traditionally have thought that influenza is a bird virus that goes from birds into pigs into humans, right? That's still true, but it turns out that relatives to influenza virus are found in many other organisms, including fish. So this is a tree of the Articuvirales. So flu is over there, and near it now we have fish, we have hagfish. They're also jawless vertebrates. So again, that suggests these are ancient groups. It's not as recent history that you think. And Mary Petrone in my group has recently been looking at Cnidaria. Now, Cnidaria are one of those phyla I showed you at the start of animals that are complete and never sampled. And they include things like jellyfish and coral. So in coral, in Australia, we have a big coral problem, coral bleaching problem. So I've sampled lots of jellyfish and lots of coral. And they have, it turns out, these Articuvirales in, these animals. So jellyfish carry flu-like viruses, right? So that puts you can ignore this, that puts the age of the influenza lineage maybe 600, 700 million years old, right? These are ancient viruses. So that's the Articuvirales like flu. The same is also true of the flaviviruses. I won't go into any detail. Flavivirus, things like dengue, yellow fever, Zika, very commonly infectious. Again, we've sampled these Cnidaria, these invertebrate phyla, and we can find flaviviruses in these species that suggest that they're very ancient, maybe dating back 750, 800 million years, right? So these are ancient things. Okay, so these viruses they're everywhere, they're very diverse, and they're ancient, right? They also exist in ecosystems. So we normally think about viruses in context as causing disease, but I sample lots of animals, and most of the animals that I sample are actually not sick at all. They're completely healthy, and they just carry lots of viruses. So humans are actually very unusual, they're very clean. My group don't want to work on humans ever, because they never find any viruses, okay? Animals, wildlife carry lots more. I think they exist in a kind of complex ecosystem. Now, good evidence of that is, in fact, SARS-CoV-2. So SARS-CoV-2, Marion may know more than me, but Of all the viruses that I've ever worked on, I've never seen a virus that's managed to jump to so many species as SARS-CoV-2. It's just unbelievable. And humans, the key thing is that it's come from an animal into humans, and humans will also pass it to many other animals too. This is a great website from a group in Vienna that did this. And it's just showing you on a map of the world. This is a map of the world, just showing you the different animals that have picked up SARS-CoV-2 from humans. Okay, so you can see here we've got lots of mink, we've got dogs, cats, lions, tigers, white-tailed deer in the US, unbelievable prevalence. Nothing yet in Australia and New Zealand, kind of suspicious. You can't see those down there, actually. I think the lights aren't dim enough. But so it's an amazing diversity. So humans are key vector species in ourselves. We're passing to other animals. And I think what that tells you is that when we think about disease emergence, we normally put humans at the end of the chain, right? But that's not correct. Humans are actually part of an ecosystem, and viruses live in ecosystems, and they're continually jumping between species. And if you look at wildlife, they contain lots of viruses. And a really good example of this, and the animals I work mainly on, I'll do this very briefly, are fish. Okay, we have a lot of fish in Australia. And my group get to go to nice places to sample them. So this is Northern Australia. This is the Great Barrier Reef that I'm sure you've heard of. And like I said, it's going through severe bleaching problems at the moment as water temperatures rise. What we've been doing is been going to various places in the Barrier Reef. So it's a massive coral ecosystem. Two particular islands, one called Orpheus Island and one called Lizard Island. And we take these bits of the reef and we sample all the fish and it's like finding Nemo, right? There's so many species in one little reef. And we sample all these kind of reef fish, and they carry a ton of viruses. I won't go into this in any detail, but even some of these fish are absolutely tiny things. So there's a thing called a pygmy gobby, which is the size of my thumbnail, and that lives for about 60 days. They carry lots of viruses, right? So these reef ecosystems are absolutely, this is just, all these pictures here are just showing you the kind of diversity of viruses in these fish, and there are lots of viruses in this ecosystem. Now, what's interesting in this particular Great Barrier Reef ecosystem, the viruses aren't actually jumping between the fish very often. The fish come from different families, different genera, different orders, and they don't actually share that many viruses. There's lots of viruses there, but the fish species tend to have different ones. We did actually, one little thing we found is that, interestingly, small fish had more viruses than big fish. Quite why that is unclear. The small fish don't tend to live very long, so maybe they devote less time to immune defenses, and maybe that's why they can carry more viruses. But it's quite interesting bit of ecology. Anyway, so there's lots of fish in these ecosystems, and they're highly diverse. I won't go on to that. Now, we've also just done, so that's a marine ecosystem in Australia. Okay, lots of viruses, but they don't tend to jump between them. What we've also done just recently is look at one of the best stories in the whole of evolution, and that's the cichlid fish of Lake Tanganyika. So this is a very famous story in evolution biology. So these fish entered this lake is in East Africa, okay, and the fish entered that lake about 10 million years ago, and they've diversified massively in Lake Tanganyika. It's a really textbook example of adaptive radiation. So there's something like 240 species of these cichlid fish, and they're different tribes, and you see that how much they vary in body size and shape and color. And lots of people are interested to know how that actually happened. What we have done is sampled the viruses in these fish. Okay, so there's we're down here obviously, and there is Lake Tanganyika up there. So these kind of finger lakes. Now, we also sampled these fish to see what viruses they carry, and they carry an enormous number of viruses. So and no one's thought this before, but this classic evolutionary organisms, which have diversified over 10 billion years, have also diversified in all the viruses they carry, and they carry an enormous number of viruses. And what's so interesting is, unlike the reef system, in these fish in Lake Tanganyika, they're jumping between them absolutely all the time. And the reason why, probably because they're actually very closely related. They've all diverged over about 10 million years. So whereas in the Barrier Reef, you've got fish from very different taxa who diverged millions of years ago. Here, it's much more recent evolution. So all the fish are pretty close related. And the viruses just jump between the fish all the time. Okay, it's a very kind of interesting system. Okay, so that's the ecology. Now the last part, and I have no idea how long left. The last part of my talk, we're running late, by the way. Okay, so you know I've got a few minutes. So the last, I'm going to talk a bit about the origin of SARS-CoV-2 because this is just never ever goes away, and it's been, you know, it's been a ride. Okay, so if you actually look online, you see some stuff like, you see lots of conspiracy stuff. You see lots of these kind of flow diagrams that don't kind of make any sense, like science. That's us over there. You get these kind of people showing that various papers and all the linked authors and how we're all kind of like corrupt and stuff. Here I'm accused of faking data with the Chinese military. Here I'm faking pangolin sequences with these Chinese military agents who are so subversive they stand by their post at a conference advertising their work. They didn't think that one, did they? Here I am, and that led, in fact, to a headline in the Australian newspaper about the this is about me, Red Army virus probe, okay, about my faked pangolin virus sequences. Extraordinary stuff. Have you seen this, Marion? So there are lists of these going around of these Communist Party scientists. I'm happy to be on this along with Marion. It's a great list of look at these evil people on that list, right? It's just extraordinary. Here I'm being compared to a well-known British comedy character. We have the same initials in our name. And my favorite was just the other day, actually, where someone on Twitter said, I was like Oppenheimer without the security clearance or the regret. I thought I was more like Barbie Ken, so I was actually a bit disappointed when I saw that. So, you know, this is it's just absolutely endless. So what's the truth of this, right? And just ignore that, really is just conspiracy theory. So at the time of writing, the closest viruses we have to SARS-CoV-2 are in animals, and they're actually in horseshoe bats, rhinophilus bats, which are very common in Southeast Asia. And at the moment, and there'll be more coming, the closest ones are from Laos, okay? And they're called the banal series, and that banal means bat anal, by the way, okay? And so they're about they're 96.8% similar in gene sequence to SARS-CoV-2. Now, it's quite hard to work out exactly how divergent that is. Philippe Lemay will give me a good answer, because there's lots of recombination going on. But it's a few years of evolution between these viruses and SARS. There's a gap between these viruses of a few years and SARS-CoV-2, so we're still missing that. And they're extremely similar to SARS-CoV-2, particularly in the receptor binding domain. It's a true receptor binding domain. In that receptor binding domain, they are almost identical to SARS-CoV-2. And these viruses exist in nature, so they could emerge again. This is the functional core of the virus, and bats in nature carry viruses that are very much like SARS-CoV-2 that can bind the particular receptor that these viruses use, human viruses, very well, right? And these bats are very, very common across Southeast Asia. This is a heat map showing you where these rhinophilus bats live. And you can see over here, the kind of darker the color, the more there are. And there's lots and lots there. So as we sample more, we will find more and more of these viruses. And there will be absolutely thousands of them out there. And a large number of them will be able to infect human cells, I would think. So that's kind of where we were till very recently. Mang, whose picture I showed you a little while ago, recently we did some sampling, or Mang did, in Yunnan province in southern China. And we sampled some bats there. And sorry, it's a bit technical. Basically, we found one virus that was a recombinant between a SARS-1-like and a SARS-2-like virus. And in the SARS-2-like bit over here, it was as close as any existing to SARS-CoV-2. So its receptor binding domain, that kind of functional core, is very close in these viruses from Laos, was even closer, or as close in this particular one from China, from Yunnan. So there will be lots of these viruses out there in nature. We've not yet kind of found the closest one, but it's there. So then how do we get from these bats to Wuhan? That's where it started. What's the link? And I really do think the link is this particular market, this Huanan market in Wuhan city. So work led by Mike Warraby last year. What we did, we took all the first cases of SARS-CoV-2 in December 2019. Marion was involved in collecting these data, and we put them on a map of the city. Okay, and then you look, and what you say is, okay, given that distribution, what are they clustered around? Is there any particular thing that these early cases from December 2019 cluster around? And it turns out they all cluster very closely around this particular Hawaiian market, okay, which is right there. And the most interesting thing was some of these patients were linked to the market. They worked there, they shopped there, that kind of thing. But what's so cool was a lot of the patients had no links to the market at all. Okay, there was an early case of COVID-19, but both those groups were linked spatially around the market. So that really was the epicenter. And a lot of people said, oh, it's just sampling bias and they reported cases preferentially for the market. That's simply not true. We've done a lot. What this picture here is showing you. In fact, this particular market is not a very busy market in Wuhan. There are lots of other things that are busier than that. It's not so many people. It wasn't preferentially sampled. This really is the epicenter. And the other thing about the market, and I've been to this market. I was there in 2014. I took some pretty bad photographs. If I'd have known how important they'd been, I'd taken better photographs, I have to say. But I was there in 2014, and I saw wildlife in there. I took a photograph of a raccoon dog. These are from 2019. Okay, that's a marmot, that's a porcupine, that's a raccoon dog. Okay, and they were in the market. So wildlife was there, and some of these species are the same species that were infected with SARS-1. That's the critical point, and they were in this market in Wuhan at the right time. And then what's even better is that when the market was closed on January the 1st, 2020, the Chinese authorities came in and they swabbed surfaces. They swabbed kind of gloves and door handles and cages and drains and bench tops, and then they were later tested for the presence of virus and sequenced. We've now analysed those sequences, and what you find is most of the market's in kind of two halves, a west side and east side. Most of the positive cases are in the kind of southwest corner, and that's the corner where the wildlife was. And if you look at the sequence data metagenomically, the animals that were being sold there were things like, you can read it yourself, raccoon dogs, civets, porcupines, exactly the same species involved as SARS-1. Okay, and amazingly, there's one stall there that's kind of like the most positive stall in the market with cases, and that's the same one that I photographed in 2014 that has raccoon dogs in it and some birds in it too. So to me, it looks just like SARS-1. SARS-1 involved an animal market in Guangdong province in the end of 2002 involving civets and raccoon dogs, and here we have an animal market in Hubei province in 2019, and their raccoon dogs and civets were there, and other species too, right? So that, to me, is as good as we're gonna get. Now, so I'm almost up to 5 minutes. So why then do people keep saying it's the lab? Why do people keep saying it's virology? And I want to tell a little story here. It's my favorite story, great story from history, and that involves what's called the Umbrella Man. Now, so what happened, one of the most famous events in history, bear with me, you'll see why I'm going to link this. The most famous event in history was the assassination of John F. Kennedy on December the 22nd, 1963, right? So it's a sunny day in Dallas, Texas. It rained the day before, but this day is kind of warm and sunny. In the whole of Dallas, there's one person holding an umbrella, okay? See that? And he happens to be exactly as the motorcade comes in a Dealey Plaza and the bullets are fired. The only person in the whole of Dallas holding the umbrella is this guy here, exactly as the shots are fired. So how can that not be? How can that not be important, right? It has to be. How can it not be? And many people thought it was, okay? And so they actually suggested maybe he was the assassin, okay? So his umbrella, as he opened it, it fired a little flechette. You see that? It fired it across, and that's what hit Kennedy at one point. And people, there's loads of conspiracy theories saying somehow this person must be linked to the murder, because how can it not be true? So then they actually, in 1978, they did an investigation, they put a call out to find the Umbrella Man. Who was it? Why were they doing it? And what it turned out it was, this person was making a protest against John F. Kennedy's father, Joe Kennedy. Now, Joe Kennedy was the ambassador to Britain in the 1930s. And Joe Kennedy supported Neville Chamberlain's, the British Prime Minister's, appeasement of Hitler before World War II. And Neville Chamberlain, the British Prime Minister, was always photographed carrying a black umbrella. So this was his silent protest against that. And the point of this is, you can never, ever, things that look really strange, you can never think of the real explanations for it that are even stranger you can ever imagine, right? And the word is virology, it is the umbrella man. It happens to be there, okay? But that's all it is. There's no more than that. And there's lots of other information. I have no idea what time I'm meant to finish, actually. There's lots of other information. I won't go into any detail, but if you actually look at the lab theory, there's simply the lab origin hypothesis. There's simply no evidence for it whatsoever. You can kind of go through the flow chart. Most of all, there's simply no evidence that the virus was ever in that lab before it started. That's the key thing. If you can put it there, I can believe it, right? But there's simply no evidence it ever happened. There's a very good email there. I'll discuss that another time. Now, the last thing I'll talk about 2 seconds is, so, you know, I'm trying to link the 2 parts of my talk. What I did in the first part was show you how virome sampling is really important. And the second part, I talk about COVID-19 origins. And the 2 things are completely linked. And what the market tells us is that this trade in farmed animals, in wildlife farming, and also fur farming, because some of those animals are fur farmed, is actually extremely dangerous. And one thing we did in 2021 was go and sample various animals at wildlife farms in China. The animal markets are closed, but the farms that supply the markets are still open. So they're selling things like raccoon dogs and porcupines. That there is a menu, a live animal menu you can find at these animal markets, okay? That's a menu you can find, okay? These are animals that are being sold there. So we went to these farms and we sampled them. And they contain lots of viruses. These animals have lots of viruses, including many we think could jump species boundaries and affect humans, okay? So things like H9N2 influenza virus was in these things. Oh, by the way, in that market, the Wuhan market, when they sampled it in 2020, we've now found that H9N2 influenza virus was there as well, okay? So it wasn't going to be SARS, it was going to be avian flu, right? So these animals in these wildlife farms are full of virus. I'm going to show you a little video of a few of these things. So this is an Asian badger. And if you watch this, do you see its nose? It's very slow. The internet's not very good, but its nose is dripping. That animal has avian flu, H9N2. Right? Sorry, it's not really. It's a bit slow. This is a raccoon dog. It's coughing. You see that? A raccoon dog with a cough. That's your worst nightmare, right? That's got a respiratory disease. We don't know what that actually is. We also found other raccoon, and I apologize for this video is going to come, okay, so don't. This is a raccoon dog with a gastric infection. This was actually an alpha coronavirus. I see people handling them with no gloves, right? So this is what causes pandemics. This is it, right? Not labs in cities, it's this. And this industry is absolutely enormous industry in many countries, this wildlife and farming industry. And the same thing's happening now with H5N1 flu. Okay, Marion knows more than me, but it's now getting into into a variety of birds. It's also getting into farmed animals like mink. Okay, and also raccoon dogs and a variety of other things. Here are 2 papers just recently published in it's one from Finland, there's also one from Poland that's happened very recently. So the same thing's going to happen again, right? It's exactly the same animal-human interface that's getting narrower and narrower, or wider and wider and wider. And I'm going to stop there, I think, just to kind of on a grim note. So, but the good news is that metagenomics gives us a really good way of actually identifying all these sort of things very quickly. So I think I'm very slow actually on that. That's it.
Given that we are a few minutes late,
questions?
I would like to take 10 minutes for questions, because I think that's a
okay.
whole lot of questions there.
Okay.
Yeah.
I wasn't sure what time I was meant to finish, but anyway, questions? There's one there.
Yeah, thank you very much, Prof, for that interesting talk. My question will go to So I know people are now working more on switch samples, especially in terms of surveillance. I don't know what's your take on this, but this is something that I'm really, I thought maybe if you're talking about animals and then, jump of viruses from animals, and people are now working on more, maybe not more, but this is what I see now, that people are really interested in sewage samples, trying to get samples from sewages and stuff. So what's your take on that?
I might let Marion answer that question, because Marion knows much more about wastewater sampling than I do.
Happy to discuss, but yeah, it's clear that with SARS-CoV-2, unlike we had expected, wastewater turned out to be a very good tool for surveillance, and there is studies that are exploring that for other, just looking at the entire virome, also looking at AMR genes. So I do think, certainly for big urban areas, that is quite interesting to explore, including the dark matter in there, so the stuff that we don't know, but it could actually be a really interesting tool to explore.
Yeah. One thing is I do a lot of animal sampling, and we tend to sample fecal samples because it's non-invasive. But I do also I work with vets who sample post-mortem, okay? And so from that they can get other tissues like liver and brain and blood, whole blood. And for example, I've sampled bat tissue, so diseased bat tissue, okay? And what you do find is the fecal samples, they're interesting. You see different viruses in the fecal virome compared to the tissue virome. So some coronaviruses are quite easy to detect in fecal samples, but other viruses we only ever see in tissue. So you will get a slightly different biased view of the virome depending on what tissue you sample.
Working. Okay, thank you very much for the lecture. What do you think are the most promising avenues of research with the big data that you've uncovered through metagenomics, when we have like hundreds of thousands of new viruses? How do we make sense of it?
Yeah, so I think you'll see more approaches trying to do that using structure, okay? So we use the polymerase, but people try on other genes as well, so the envelope genes and the capsid genes. That's going to happen. The really difficult thing, actually, is you can detect the viruses using structure, and you could confirm that using some laboratory means. You could do micrographs, and you can see them. The problem is you could never include them in an evolutionary tree because they'd be too divergent. This is a completely different problem, and so what we need to have is this is a kind of technical thing, but it'd be great to have a way of doing phylogenies based on structure, okay? Because you won't be able to align these sequences to anything you've got because it's too divergent. So quite how you depict evolution is going to be very, very difficult to do, I think. So you'll see lots more virus discovery coming though very soon. In different environments, in different tissues, animals, whatever.
Thank you. You opened a whole new world of aquatic viruses for me. I was wondering, did you already see evidence of jumps from fish to humans? And do you think, from a One Health point of view, that fish are an important reservoir to study more?
The first bit of fish virus stuff that I did, I went to the local fish market and I bought a load of fish. And we found a load of viruses. We ate the fish afterwards. We barbecued them, and we never got infected. So normally, I don't know any example of a fish virus infecting a mammal. You have one exception, a second, right? Because their cell receptors are so different, and they just don't bind. That said, if you look at, so I don't think they're a risk, okay? So if you buy a fish, and don't worry if it's got a virus anyway. That said, if you look at the hepatinoviruses, hepatitis B-like viruses, there has clearly been a jump from fish to mammals at some point in evolutionary history. It's clearly happened, right? So it's not impossible. It has happened at least once, but on average, I think they're pretty low risk. The way I think about fish is that they're an awful lot of fish, right? And they're extremely diverse, and they're ancient organisms. And so they are a really good way of understanding the virus ecosystem, the virus universe, okay? And you can work with fish, and people ignore fish, but they just carry an enormous number of viruses. I think it's a truism that every viral family that people thought was mammalian, you can find in fish, right? Filoviruses like Ebola, they're in fish. Hantaviruses in fish. So that tells you that these things are ancient, okay? They're all as old as the vertebrates, easily.
So you made an interesting observation that there's evidence for a lot of reverse zoonosis from human to a wide variety of different animal species. So are there new reservoirs that we've created for coronaviruses that we should be worried about?
Oh, crikey. Well, this is a really, Marion and I. So look, clearly the virus is now established in other taxa, right? The question is what that means for the future, right? And you could argue it in two ways, right? So one is you could argue this is an evolving reservoir that could re-emerge in the future, certainly possible. Or you could argue that as it adapts to that species, it'll become more deer specific or more mink specific, and then less likely to jump back into humans. Because that's what happens in evolution of any virus, right? And we're not at that stage yet. So in the short term, I think they are probably a reservoir that could for re-emergence. In the long term, evolution should take them away from human specificity, right? So, but we're not there yet, though.
Thank you very much for a very interesting talk. Turning it around, could we use the marine viruses as a mechanism for scoping defensive mechanisms? So how the plants and fish and sponges have their own chemical protection against some of those viruses, which could lead into human treatments or solutions?
Yeah. So I'm going to answer a different question. So as I said, one of the striking thing is every time we sample wildlife, and I sample lots of wildlife, you find viruses, and these animals look healthy, right? I mean, the fish look fine, we ate them, they tasted fine. We sampled birds, other mammals, reptiles. I mean, they carry an enormous virome, and there's no obvious overt disease. So the question is, why is it? Okay. People in the mammalian world are obsessed by bats. I have found viruses in bats that I think are causing illness, by the way. I don't think bats are not magic. Bats can get sick. But generically, there is an important question: is why some taxa can carry these viruses, and with no obvious disease syndrome. And the way I see it now, having done so much of this kind of big stuff, is that most viruses, in fact, don't cause disease, as far as I can tell, right? And that's actually, as disease is a lower common occurrence in most cases. Why? I have no idea.
So,
well, no, but sorry, the virulence question, yeah, that's a very difficult one. And I'll give you a very brief. So he says basically you don't want to kill your host. So in Australia, we released 2 viruses to kill rabbits. Myxomatosis, myxoma virus, that declined in virulence. They're still really virulent, actually, still kills half rabbits. RHDV, a clade C virus, that's increased in virulence through time, right? Because, in fact, it's passed on by blowflies who feed on carcasses. So it pays to have a dead rabbit because then you get more flies that transmits it. So it's very hard to predict the evolution of virulence. It depends exactly on the mode of transmission.
So yeah, thank you very much, Adi. We are so excited that you accepted to come here. Yeah, and not only your talk is absolutely phenomenally scientific, but many of you that work in the field will know that was Ed Holmes that also did the first or released the first genome of SARS-CoV-2, and without that we would not have identified the virus, or you wouldn't have diagnostic or vaccines done so quick. Yeah. And the main thing that always impressed me about Ed Holmes is that his scientific level is just sky high. I think that he has over 100 Science and Nature papers. But the way that he's so humble, and not only that, all the excitement that you have for science, I think that you have been inspiration, not for me, but for many of us. So thank you so much, and I think that he more than deserves a very large applause. No, I'm going back. Sorry.
All right. So my turn. I'm still trying to shake the image of a sneezing jellyfish with a flu virus. But that's actually quite a smooth transition to what I want to talk about, which is how do we actually interpret the findings in connection to health and disease. And I titled my talk, Getting Most Out of Imperfect Data. And that's also from quite some years of interactions, collaborations with data scientists who tend to, you give them your most precious data sets and then it's like, well, it's a little bit meager. Do you have more data? So, hey, this is your reality check. So I also think it's time to really think about where the new technologies that really have exploded and gained central stage with the pandemic, particularly genomics and the data analytics, where that will take us. And this is an image that has been developed by a guy named Gartner, who said there's a technology hype cycle, and he used that to really study the long-term potential success of new technologies. And there he described that there's always this phase of, wow, everything is going to change. We have this new trick now. We really will solve all these questions that we were not able to solve. But then very often, more often than not, that's followed by a dip when it turns out that some of the questions really still, despite the new tools and technologies, remain very difficult to answer. And I think that's a risk that we see also with the genomics and the data analytics, but really, I think our joint interest and responsibility is to try and not have that dip in expectation, but really to build this expertise carefully. This is also when looking at the extent of sequencing that was done during the pandemic. Massive, crazy at times, I would say, incredible amounts of sequencing, and this is data in the public domain in the nucleotide archive, where there's now 6 million raw read sets that all have been processed with one bioinformatic workflow, and that leads into data sets with billions of mutations that all could have an effect. All could have an effect on the virus phenotype. And yet, if you look at some of the deepest studies that are done on this diversity biologically and experimentally. That's work, for instance, by the Bloom lab that does deep mutational scanning. It really only studies a tiny fraction of all of those mutations. So what on earth do we do with all this data, all these mutations that may or may not have an impact on the functions of these viruses? So there's a lot of data out there. There's also an explosion of tools and ways to try and look at the data and make sense of the data, and having charts and having variant maps and in different shapes and forms, different names. And here I think this is the poor public health guy throwing up his hands in this case, saying, okay, I'm going to go to the liquor store, I'm done with this, I don't see where to go with this. I think this is a pattern that I certainly recognize nowadays when we are, just this week again discussing variants. I even cannot remember the numbers anymore, but this one is B286, right? That's the one. How do we do that? So what I would like to take you through is some of the thinking that I think we really need to get back into the equation, and that's really what defines actionable information. That's the thing here on the right. So eventually, if through all these scientific endeavors we want to generate and create actionable information, that means we really have to think through this whole pipeline of steps to come up with that. So, because first of all, what is the question? And depending on what question there is for a specific study, the sampling needed may be very different from other questions. So what to sample, where, when, how much, and also, do you need to take controls? What do you compare it with? If you take, for instance, samples from a diseased animal for metagenomics, can you also collect samples from healthy animals to see if there's a disease association, as an example? So then once you've done that, there's also important, depending on the question asked, how to actually analyze the samples, with which tool, which method. They all have their pros and cons. And what is the quality process here? Also very important because of some biases that may be introduced. And eventually, with the data, with metadata, with the sequences or other lab readouts combined, then you move into analytical workflows. Again, the choice of which very much depends on the data and the questions you're trying to address. And I will go through that in working through some examples here. So here's the first case. I'm a vet, but I do work in a medical hospital. I still can fool people a little bit because I can, from my veterinary education, throw in some medical terms, and that. So I'm somewhat expected, somewhat tolerated, I would say. But here's a patient, 72-year-old male, hospitalized, severe diarrhea. And when the clinician asked him, it turns out that he also had 3 friends that came down with something. So I want to hear just a couple of ideas of what would you want to know and what would you want to sample. Just shout. So where were they? What they ate? Timing of what? Yeah, okay, so when was the contact? What were the connections? How old are they? Yeah, so all of these questions, right? So yeah, let's have a look. So it turns out this person was infected with a norovirus. So if I look at that, my first question would indeed be, could this have been part of a cluster? Because we know there's noroviruses that are out there, and this is a tree, a phylogenetic tree of the norovirus diversity. There's extreme diversity family Caliciviridae, there's two main genogroups here. 80% of outbreaks are caused by viruses from this genogroup, 20% by viruses from this genogroup, but there's also all of these other diverse viruses, some of them in animals. And I saw in Adi's tree that I need to look up the cell paper because she had a norovirus in there as well, and some creature that I haven't seen yet. So highly diverse, so I would want to know which norovirus. Is it business as usual noroviruses, which is this little branch here, or is it one of those weird ones? But that's because I'm interested in noroviruses. But I would also want to know more precisely what viruses these are, because the different viruses in the different lineages differ. They differ in severity, in transmissibility, in host binding, and in environmental survival. And some of the evidence for that comes from a global collaboration that has been up and running for since the mid-90s, where labs and colleagues all across the globe share data, sequences, and some epidata on outbreaks of diarrheal disease into the NeuroNet database. So because of that, we know and we can compare these behaviors of the different viruses. And from that, we've learned that some noroviruses, the G24s here, really most often are transmitted from person to person. So these are really the outbreak viruses. That's what you find, where you find them. And you also see that if you look at the settings where you find G24, that is in healthcare settings, in hospitals. So if you first thought, if you have a story like this, a patient, 3 friends, would be, well, it's probably one of those common genotype 24 viruses. If you look at some of the others, then you see other modes of transmission popping up, like foodborne transmission, much more common, for instance, with the genogroup 1 noroviruses. Now, if this person, from the questions that a clinician may have asked, which clinicians rarely do, what did you eat, then maybe they had a joint party and maybe they had shellfish. Very common cause of disease. And why is that? Because they grow in our environment, in oceans or coastal areas. And we tend to throw a lot of stuff in there. There's wastewater, so you may be surprised to hear that it's actually very difficult to find coastal areas that do not have presence of human fecal coliforms. It's very difficult to keep, well, shit out of the food chain, literally. There's also animal waste that may flow off into the coastal areas. So you start to see a picture here, very nice mixing. There are sea mammals in that mix that may also deposit their own viruses. So, and that is picked up very nicely by filter feeding shellfish, because these are wonderful creatures. They filter the water that they live in. An oyster can process about 200 liters of water and retain whatever's in there. And there's for some oysters, the ones that have invaded Europe, for instance, we know that they can actually retain noroviruses because they have the same receptors as we do. So there's not only the processing, the filtering, but then also the active binding of viruses so that it nicely gets onto your plate. That's what oysters do. So then, if that were the hypothesis, I would want to maybe test oysters. Now that's a field that has been expanding a lot, and there is groups that really are expert food microbiologists that have worked out ways of trying to find viruses in these shellfish. That's by no means easy. You have to dissect oysters, you have to take out their digestive system, and then that goes into an RNA extraction, and then you can process that for sequencing. But it's also then important to contact maybe the oyster producer to understand what happened with the oysters before they were at the party. Because different things are done with oysters. Here you have first here's a site which has oysters, and you can see from the testing, the gray, the lighter part here is oysters that test positive over a certain threshold. So 70% of that batch has a lot of norovirus in them. So a highly contaminated batch. But that batch was also put, harvested and put in a different site, a cleaner part of the ocean. And you see that rinses out some of the contamination. And then before actually putting them in the market, quite often, if oysters are harvested from a contaminated area, they are depurated, which is in essence put in clean waters to allow some flushing through. So there's depuration, short-term, long-term depuration, and you see the amount of virus in the oyster going down. Now you are looking at this, you're testing this, you're finding some viruses by PCR. The health authority will ask, so is this now still a problem? Here, these low amounts of virus. Then you have to go back to what do we know about the virology here, and it is that only a few particles are enough to get someone infected. So yes, even low-level contamination will still be a problem. And I'm explaining this just to see, so as you notice that you walk through different disciplines that you really need to answer these questions efficiently. Okay, then someone will say, well, really, I want to have the sequence of those viruses to make sure that we can compare them. And that's, of course, what we all would want to see. Here, looking at genomes, trying to recover genomes out of oysters. This is work from a group that we collaborate with a lot. IFRIMER is a reference center for shellfish testing in France. You see that they can pull out norovirus genomes out of wastewater, out of oysters that they artificially contaminated. But then it's more difficult to get whole genomes out of naturally contaminated oysters, although this is already a big step forward compared to the previous methods that have been used. I would also want to know what exactly is in there for the reasons that I just mentioned. So the ocean systems, of course, do not only contain oysters and human and animal waste, but there's also the free-living mammals. And here, this is a porpoise that happened to be infected with its own norovirus, which you can see here that is staining of the intestine, so it's not a passing through, it's really an infection. Here is where it sits in a phylogenetic tree, where here are the human noroviruses, and these sequences also have been found in oysters. So with the oyster, not only you can get a mixture of human viruses, but also, let's say, a sea mammal virus. And why is that then something that would interest me? That is because we see in the norovirus field a lot of recombination. If you look at the current diversity that we know, there's actually a lot of mixed genomes, recombinant genomes, and in these viruses that linking together of genes from two parent viruses is almost always done at one specific spot here, and you can actually recognize them if you test samples or people by a PCR that uses two targets, one here and one here. That's been a routine method in the clinical field for some time. And of course, recombination is something that requires mixed infection. So you need to put two or more viruses into the same individual, into the same cell, to allow for that mixing to occur. And where does that happen? Well, that happens in situations where the opportunity for these multiple exposures are high. So that's in daycare centers, maybe in immunocompromised patients that will be infected with one virus and be super infected with a second, and also in with the animal exposures and food exposures. I would also age, someone said age. I would want to know age because of possible immunocompromised state. That's something that really became very clear also with SARS-CoV-2, that people with impaired immunity may not be able to clear a virus infection the way healthy people do. And what we've seen is with SARS-CoV-2, but we've seen also already some time ago with noroviruses that these relatively innocent viruses can infect, let's say, a transplant patient and continue to circulate. They cannot clear them and then gradually evolve. That's what these viruses do. So what you're looking at here is, again, a tree. These are some of the common genogroup 4 sublineages, and what these long branches sticking out are noroviruses in chronically infected patients. So they really drift away from the viruses that they originally were in these patients, and that also evolves into viruses with different properties shown here for this group of immunocompromised patients, mutations in important antigenic epitopes and immune escape. So we see that. So these are all different reasons why you would want to study this. Is this, the first question, is it just a cluster, a business as usual? Has this person and his 3 friends maybe had some contact with young kids, and it was a regular outbreak that you really don't necessarily bother to investigate, or was there something deeper going on? And depending on that, you really would need to design your sampling. So now we have, let's say this person has been tested, there has been a sequence generated, there is a question. Let's then compare this with a global database, right? Now that's a tragic image. This is from 2018. It's probably a little bit better now, although not much work in this field will have been done during the pandemic. But what you can see is that there's actually very few full genomes here. Unlike we've learned to do for SARS-CoV-2 and a little bit for flu, for instance, very, very little full genomes, but what you see is all these tiny snippets of genomes that reflect the different choices in different regions, the cultures of testing. So what these are sequenced PCR fragments that people have used for detection, for instance, in oysters. And that creates a sequence, but you can see if you want to then compare this to the whole global diversity, it's very difficult to do because there's no alignment here. So reason here to really also invest in broaden the sequencing to other pathogens that are also relevant, but very much less studied. So I'm showing this case. So this is really a common disease. It's one of the most common infections that we all get during our lifetime. It's a virus that shows all the things you can learn about viruses with evolution, through recombination, through drift. It shows that viruses can move between humans and animals. It shows the recycling through the food chain. Lots of things to learn there. And particularly the message here also, it means that if you really want to understand what these viruses do, you need a whole cloud of different experts and expertise. And I think that's really critical, that we really learn to break the silos and work with all these different disciplines to try and really grasp what our favorite viruses do. Plus, here the message, even a simple outbreak investigation for a common endemic disease can be very challenging. And if you're honest, this is, I think, my favorite the topic with the origins discussion. What you do see is now the world is, you know, jumping into how much evidence do you really have? And we're all used to not having complete evidence, but saying, okay, given everything I see, I conclude that this is the most likely. Now, definitive proof is extremely difficult to get. Even for these foodborne outbreaks, for instance, definitive proof of a negative is even more difficult to get. So, and this is important, and I think we need to convey that better also to the wider public. Okay, shifting gears then to making this picture even a bit more complex, and that's when we move into the emerging disease field. So investigating emerging disease outbreaks, you even want to expand further, bring in further areas of expertise, and think through what would we really need in terms of samples and data to answer some of the key questions we're trying to answer. And for that, I like to think through the different steps in a disease emergence pathway. So there's the first step. We know many emerging diseases come from the animal world. And that is a process that really is influenced and driven by things we do, by the impact of us humans on the environment through large-scale deforestation because of growing population that we want to feed with animal protein. We build farms. So that's that first step, spillover. So if you have questions there, that means that you need to focus on sampling that can address those questions. Then the second step, if you have a spillover, is amplification. And that's where we look at drivers for disease spread, like global travel and trade, and the opportunities created for transmission in, for instance, in mega cities that have grown so fast that their infrastructure has not kept pace with those developments. And then the third step is really trying to understand impact, and that brings in what population, what does the community look like? Is there immunocompromised individuals exposed? Is it people with low income that are at increased risk for all sorts of impact? So again, different conditions to consider. So depending on the question, in the first instance, if you look at the spillover question, it's important to think of farms, for instance, as multi-host environments. Do we go out and just sample the farm animals, or do we broaden that? Do we take that ecological approach in our thinking, including, for instance, environmental sampling? If you think about probability of spread, that's where you need to start thinking about travel hub surveillance. There was some interesting posters on that here in the conference, but also maybe the wastewater, the question that was mentioned there, because that's really relatively easy samples that do show what may be circulating in a whole community. And if you think about answering questions about potential impact, you really need to be set up to understand impact in different types of the population pyramid, which is very rarely done. Now, showing some of that in our pandemic experience here. So this is taking you to the Netherlands, March 2020. This is a week after our first hospitalized case was notified. There were a couple of other detections, but not a whole lot yet. But there was also, we just had Carnival. Many people were you know, flu-ish, like we all know after big parties. But there was a sense that maybe something was not right, and it was difficult to really assess what was going on because there really were reagent shortages in our part of the globe. So what was done here is a screening study with healthcare workers from a couple of hospitals, 1,000 in total, over a weekend. And that turned out that already 50 of them actively were infected at that moment in time. So, and these were all people with just runny noses, not very sick, but there was already a lot of virus in this minimum spanning tree, so all over the tree, so many introductions that had been missed that were already seeding chains of transmission. So right early on in our country, what we had was a zoonosis meeting, because that's been an experience built from Q fever outbreaks, avian flu outbreaks, that with a new surprising situation, you really want to bring experts from these different disciplines around the table. And that was also done to then discuss, okay, what do we know? Could there be something going on with animals as well? So early info here, that's February or March 2020, there was mention of infected cat that had been shown by colleagues from Wuhan. So there was something flagged, and that's of course pets, and people worry about their pets, so there were already a lot of questions about that. But there also was information from experimental infections, which had shown that several of the farm animals like cattle, pigs didn't seem to be certainly not highly susceptible, but ferrets, the favorite lab animal model, were, and that brought the discussion to potential for mink and other fur animals infection. And that is, as Eddie already said, it is in some parts of the world and in some countries a big industry, and the Netherlands was one of those countries. As you can tell here, so we had at that around that time 127 mink farms, an average 5,000 female breeding animals that have somewhere between 8 to 12 pups in 6 rounds throughout the year. So lots and lots and lots of mink produced here. And that also, that sector involved 1,600 employees. So because of this sitting together, vets, medics, virologists, it was like, okay, maybe, you know, have look out for possible infections in these animals. So sure enough, end of April, the first detections were there. And this was, if you think about that here, because of an increase in mortality. So from 0.6% to 1.2%. So what this is, if you really think about it, so you have a farm with 10,000 mink. So that means there is every day 60 dead animals. So that's business as usual. But here a farmer noticed that, hey, it seems to be more, I have a hunch. And then they submitted animals for testing to an animal health service. And they showed that these animals had a severe pneumonia and that they were indeed infected with this virus. And the reason they could do that is because they had been pulled in as a test facility to scale up the human diagnostic testing. Otherwise, I don't think this would have been detected. So then, of course, what you do is then look at what exactly is going on. And you see here the date of detection, 2 farms, top one and the bottom one. 2 weeks, a little less than 2 weeks after the initial symptoms in the animals. And then, of course, you bring in the public health authorities to try and figure out how was this introduced. Can we figure that out? So in retrospect here, through interviews, turned out that the family on farm 1 had symptoms. No one was tested, but it could have been a cluster of COVID. But in the second farm, a farm worker had been hospitalized for COVID, but no one had asked about the farm and the work environment. So that had been missed. And this is not unusual. So if you think about that, then again, you can think about what would you want to know, what would you sample. Of course, now we know much more than we did then, but I will just go over what was done here. So first of all, so this was really a One Health outbreak investigation. So first of all, the Animal Health Service was asked to really test more dead mink. And this was, it's not something you can demand, because this Animal Health Service is a commercial service that is paid for by the farmers. So the farmers really have to want to do that. And the reason why they agreed is because there is a compensation mechanism if something happens to the animals. Then the human health authorities had to be brought in to say, okay, let's have a look how much transmission there is to people. And then if you have, and also to look, are there any people in the same neighborhood, in the same postal code that are infected, and what viruses did they carry? And then we had questions on how does this exposure go, where does the air go, where does the dust go, is this a risk to humans? So studies there. And here you see the first findings that looking at the genomes from the animals in a continuing series of farms, there were actually 5 separate introductions, but also in purple here are the people from the community, and there was seemingly no, certainly not widespread community spread. So important to understand that. But if you then look at those introductions, so in blue here are the 5 separate clusters of mink farm. You see that the introductions may already have started before the first human cases. So really, that initial thinking about what is coming our way already should have been organized in a One Health way. It wasn't, but that's a lesson learned. But what you can see here also is that you have this blue to red pattern, which really reflects the virus jumping back and forth between humans and animals, and with that also picking up mutations that then were confirmed to really have something to do with adaptation to mink, as Adi just mentioned. Now then you get very worried because you want to know how big is this, and just from doing case finding, passive case finding, that's extremely difficult to know. So what was done here was a study on 16 farms, trying to contact the people that live there, particularly testing them by PCR, but also by serology. And what that told us that a majority of individuals working on those farms was getting infected. This was at a time when the seroprevalence in the community was less than 5%. So majority of people were getting infected. So that really was brought into the national outbreak meeting, and it helped in the decision to actually stop the farming completely. Now then you also get questions on, so now we have these tables full of animals churning out virus. What should we do with that? Because you cannot just get rid of it. These are fairly open environments. So this then brought in the environmental specialists, people that also study pollution around highways, for instance, and looking here at a sampling process in an infected farm stable with a person here with carrying a dust or an air collection device just to look at what is possible inhalable
air contamination, here a stationary air sampler, and here what is called electrostatic dust collectors that you put somewhere and that really capture the dust that is flying around. And here to the right, you see the dust test results, quite high viral loads associated with the stable dust. No viral loads with the inhalable air, some but not so high viral loads with the air, just circulating air from the stable. Not easy data to confer to a public health person, because how do you interpret that? Just think about it. I can share how we worked with that, but it's not easy answers there. So then the next step was to really put all this data together. So this is quite extensively investigated. This is not routine. So looking at public health investigations, looking at the whole tracking of all the movements of people and goods on the farms, trying to understand how the virus may have been brought in. Bringing in the genomics, of course, and all sorts of other factors. And what you can see here is then if you look at which of those farm clusters can you actually link, despite all of this work, you can still see there's a lot of separate clusters. So we cannot explain the whole transmission process. Despite all of that work, it's extremely difficult. There's two factors that stood out. Distance between farms, the further away, the less connections. So that suggests there is some wind or air transport. And of course, farms that had links through personnel, they had more risk of infection. But still a lot of missing links. So then another study was added in, and that was like, what else could be playing a role here? And that is looking in those farms that are considered somewhat biosecure, they are not, but bringing in building specialists, ecologists, ornithologists, and all the experts that you see here to see what else creeps in and out of these farms. And that was done by both by observations, but also by asking the farm owners. And so there was evidence of lots of different animals: cats, foxes, martens, badgers, and feral mink moving in and out. And then, of course, the bats, rodents, and very often birds that move in and out. All of them can be vehicles for transmission, maybe even be involved in the circulation of these viruses. For instance, feral mink, escaped mink, and those populations exist almost everywhere where there's mink farming, and they like to hang out around the farms and move back in just to grab some food. So they may certainly play a role in sustaining infections. So that example is meant to show really that farms should be seen as a multi-host complex system, where if you really try to understand what is going on, you have to carefully design what to sample, where to sample, how to sample, because the picture is easily much more complex than initially thought. And really assessing quantitatively how these viruses actually are transmitted is very challenging. And here's just a recent example. This is one of the publications that Adi mentioned, a cryptic SARS-CoV-2 lineage in Poland in mink farms. So this is a virus that clearly is very different from viruses that we've seen before, so also from Poland. And it's absolutely unclear where that virus came from, or where, whether that has some kind of reservoir outside of the farms. This virus has not been seen in the human patient sequencing that has been ongoing. So again, I'll trigger you to think what you would want to know here and how you would actually go about trying to address this. And it's not easy. We've not been able to convince the Polish authorities to dig in somewhat deeper. I want to close with just mentioning the huge importance of data sharing for this. So This is work done by so many different people that all have a stake. They all have invested time. They may have used grant money, and they are encouraged to share data. But what's in it for them? So that sharing of data is a critical piece of successful outbreak investigation. And but there is a continued challenge, and that's of sitting on data because of well, some interest. And I do think that just from looking at all the discussions and the poster that the VME Club is really an important driver of change of this culture. And Annemie, I thank you for starting that initiative someday, because there's so many One Health posters, collaborative studies that you see evolve here, and that's really wonderful. Here's an example also in the public domain from the COVID-19 data portal developed with European Union funding with one of the three constituents of the global shared open database for biomedical data. And that actually attracted people from more than 105 countries sharing raw sequence data. So yes, we were discussing that maybe the raw data need to go out. Please keep them in, is my plea here. Because we need to learn how to work also publicly with these data, because otherwise my worry is that we will build one portal after the other just for one community, and that's just not how viruses cross borders. We should also cross borders with our thinking on sharing this data. So I think the next and really holy grail for all of us will be, can we move towards true prediction? So and by really understanding what is going on in the ecology, as Adi said, so move away from thinking of people at the center, controlling it all, to looking at ourselves as part of a shared environment that we really share also with virus, and maybe actually learning how to predict where outbreaks occur and actually do something to prevent them. So that's what I want to leave you with, really closing off to say that it's really important. So whenever someone comes to me and says, do you have more data? I tend to yell at them now, because it's so difficult. That's what I'm trying to convey. It's so difficult to really get high quality data. We have some people here that are struggling with that, but I think one of the challenges, what can you do with imperfect data? Because that's the routine. More, if you have a perfect data set, you're perfectly happy, but also very, very, very exceptional, because that rarely happens. So work with whatever is available and try to not overinterpret, even if you have to do that to get grant money. And collaborate with data generators and people that need to interpret the data. And just closing to say that this is all really work reflecting many different people being involved in many different consortia, and I do think that's really the spirit of work that we need in the future.
Okay, so we'll take some questions. Thank you so much, Marion. This was very inspiring to me. So some questions.
Thank you, Marion, for this excellent talk, and I'm really happy that you have touched on many aspects that come in different lines. Like I liked the slide that you said from compare, where you start with the sampling, then you with the analysis, and so you look further than just identifying viruses. You look to the whole system. So I like that. But can I challenge you?
Sure.
So could you bring up that slide from compare?
There we go.
Yes, yes, this slide. Okay, so I challenge you to add an arrow. So what are we actually doing? Because you say actionable information. So what actions are going to happen?
Yeah. No, I see your point. We of course also discussed it. So here this ends at, so this was actually thinking about who is at the receiving end, who has the question. So this could be the clinician, this could be a public health person, it could be a researcher. So then that's where this ends. Of course, if there's a whole other lot of questions on how do you provide data that are actionable, which is very difficult to do. I think an example is that that work on how do these viruses transmit in the mink farm environment. And that may seem a weird question, but this is a very, very important question for the people that control these kinds of outbreaks. So, because they were not sure, do we need to do like with poultry avian influenza, do we need to start culling all the farms in the environment if there is airborne transmission or somehow wind-driven transmission? There's no data, of course, if you have a new disease. So, but the issue there is we can measure a lot, but then translating that into, well, okay, based on what we see, do that is very difficult. I don't think we can do that as scientists alone, but I do think that that's where you really need to bring in, you know, the multiple disciplines also involved at the people that need to work with whatever we provide. Yeah, so I do agree there, including social science.
Thank you for a very interesting talk. It's a good thing I don't like oysters. I don't think I'm ever going to eat them again. I don't know how to phrase this exactly as a question, but you mentioned the example of a mink farm and one family member took themselves to hospital or was hospitalized. It strikes me that hospitals and clinics are sort of concentrations, likely to be concentrations of immune-compromised people. And it seems like a not very sensible idea to take people who are potentially now patient zero with a novel virus that is just starting to jump into the human population and to immediately put them into the most vulnerable population possible to allow that kind of transmission to start happening really well. It's like, you know, into a tinderbox to kindle the fire kind of thing. Is there any sense in maybe policy changes that, you know, if you have an infectious disease or some, you know, even whatever symptoms that are more infectious as opposed to you've broken a bone or, you know, whatever, there should be a different pathway for people that keeps them initially out of that, you know, vulnerable population that could lead to bad population level outcomes?
Yeah, I'm not sure because I couldn't hear it very well, but I will repeat what I think is the question is, so if you have an emerging infectious disease and you have suspected cases, bringing them into a hospital is bringing them in one of the most vulnerable environments. Or yeah. Well, because if people are very sick, they are hospitalized, and really, they, you know, there's the hospitals. I would already be happy if hospitals consider that a patient is potentially infectious whenever they come in. This has been baffling me when we had the big West Africa Ebola outbreak. All of a sudden, all our hospitals went into full Ebola training, suit training. And it's like, that's crazy. You can have a person with an infectious disease walking into the clinic every day. This, you know, thinking about infection prevention should be in your DNA if you work in a hospital. So I think that's more the thing to do. Of course, including looking at risk triaging. If you have a ward with transplant patients, you might, like the situation we have now, you might want to mask earlier than if you have a ward with people with just a sports injury. very different risk category. So I think that's more likely. Really separating infectious or suspected infectious from all the rest, I think is very difficult. But the example here was quite typical. So for instance, we are a reference center for arbovirus diagnostics. We try to get background information. Where did you travel? Did you have vaccines for the differential, etc.? Less than 5% of the requests, diagnostic requests, come with that information. It's very difficult to get physicians to think about that type of triaging. Yeah, and I think that's what needs to happen.
Sorry, Marion, you can hear, right?
Yes.
Okay. So yeah, obviously we all are interested in that prevent step, and from our perspective here in South Africa, Europe and the high-income world in general acted as pandemic accelerators, and you know, and when we say prevent, you know, what Eddie talked about in his talk, and what you're talking about with the minks, we know risks. They're there. There's nothing being done about them. Okay, in case of the wildlife farms in China. So I'm wondering, you know, I mean, I know you're a scientist, you're not a politician, you're not in charge of the Netherlands, and you and Eddie have been under considerable attack for the work that you're doing. How do you think, but as a scientist who's done a lot of kind of engagement with the public, we can bring people on board for that prevent part? Because I see people being organized to actually make your life and my life, you know, in the same field, and Eddie's, and it's more difficult, to make your work more difficult, and to actually, you know, handcuff you to actually stop you working on the prevent.
Yeah, and I think that's one. So I think we all say One Health is an important approach, right? But a key question is it realistic? Can we actually prevent? I believe we can get much better at real early detection through capacity buildings, through looking at where are, you know, big changes, so that you can focus your early detection systems. But true prevention, is that possible? I like to think it is, but that does require really a system that also picks up signals that are very far away from a concrete disease. Think, for instance, what we are looking at in our country is we are one of the fastest warming countries in Europe. We are a delta. The whole country is a delta. So there's sea level rise that we worry about. So we have engineers redesigning the landscape, building wetlands all over the place, preferably around cities, so that they can buffer if there is flooding, etc. No one there has been considering what that does to disease ecology, to viral disease ecology. So that is actually a man-made problem, potentially in the making. So we are now seeing if, you know, how you can work together to actually rethink that redesign of the landscape, and we do that all the time. So landscape use change is one of the big drivers of disease emergence. And I think there's examples from the Nipah field, by understanding how that worked, just by looking at where are the, for instance, the pig farms spaced compared to fruit trees, and having protective devices so the bats cannot access the fruit or the date sap is a primary prevention measure once you know that disease ecology. So I do think we have to try and see if we can find examples like that. But it is very difficult to convince politicians to work on something that's not a problem.
Okay, can I add something to that as well? I think we've learned a lot in the last few years about the science of prevention, both in terms of ecology, and both in terms of the technologies that we now have, like genomic sequencing and mRNA vaccines. We've done way better. I mean, we're good at that. The problem is, I think we're in a worse position now than we were at the start of 2020, because the politics is so much worse.
Yeah.
And the problem now is that people, because of the vilification of people and the blame game that's gone on, people in the field who see an early case will be scared of reporting it because they'll get blamed. And you saw that in South Africa with the Omicron outbreak, and it's happened everywhere. And what really worries me about what's happened, at the moment, is that, so I mean, my colleagues were hauled in front of US Congress, right, for writing a paper, and people will not say anything because they'll be scared of being blamed. It's safer to say nothing, just let it happen, than say something. And so that, I think, is the biggest problem.
It is worrisome, yeah. I also have been at the receiving end, but I do think, so a question that I get a lot is, why don't you stop being in the public domain? But I do think, I'm saying, well, we need more scientists, because we need to better explain what we do. We need to better explain that science is also learning by doing, by reassessing, and that doesn't mean a mess up, it means that's how you learn. And that's, I think, a vastly misunderstood process. So, but I'm not totally optimistic there.
So just a question for both Marion and Eddie. Yeah. Because here we have a lot of young scientists, and a lot of people advanced. How do you motivate them how do you motivate your young scientists to play detective games, such as finding the origin of a outbreak mink or new virus? How do you do to keep them really excited that they put themselves there and go to the field to collect, to analyze? What you do, give them the fish to eat after, or what's your secret? Yeah, if you can just highlight like how we keep, because more than ever we need scientists to really be discovering viruses before they emerge. Yeah.
Well, I mean, we have Emma here from my lab. I think, I mean, when I heard about origins questions, I was like, heck yeah, I want to be involved. That's the most relevant question to look into. And so I think it is also scientific curiosity, but in my message has been an outbreak not studied is an outbreak wasted, because you can learn so much from even simple norovirus outbreaks. And so I think, and there's no stuff to learn every time. So I think there's plenty of jobs in the future.
It's been very difficult for me the last while, and what I see more broadly is that science is under attack, and for my group watching what's happening, I think a lot of them are terrified, honestly, because it is a scary time. But what we have to do as scientists is look at the data, evaluate the data, and just keep moving forward. It's all we can do, and hopefully the world will correct itself at some point, right? Because COVID is not the last pandemic we're going to see, right? And there could be one, and then they'll be crying out for us when that happens, right? So it's a very, very difficult time. I think it's a unique time in the history of science, what's happening at the moment. I really do. And I don't know why I can say, if my group look at me, I don't think they're very inspired. I think they're kind of terrified, quite frankly. I know, I'll be honest, it's difficult, it's difficult.
So in my group, people say, we're happy for you to do that. But so I think, so this was about young scientists. I think, that there is a lot more room to move in an earlier career phase. I think there's also something to be called for all scientists, where is the common outcry for this stuff, for Christian being interrogated in a horrible way, Fauci being interrogated in a horrible way? Where is the global scientific voice that says, quit doing this, this is not okay? The Australian Academy had a really strong statement, which I send it to our academy, and they're like, well, we don't do politics. I think that's something that really, I think that's a call to also the science organizations to say, okay, hey, there are rules of the game. You can be critical, but you cannot attack our scientists.
Yeah, maybe I can make a remark here. So you cannot expect from every scientist to be a good science communicator, and also you cannot expect from every scientist to want to be harassed like that. So I was involved in early communication about COVID, and because of harassment, I just said, no, not for me. I don't want that. But still, you can do something. I think we need to explore as scientists more this interface with politicians, and we don't need to become politicians, but some of us, not everyone, but some of us need to be able to talk to politicians, need to be able to talk to the public. And so I think this is a skill, a new type of skill that we should give to our virologists, because otherwise this direction that the society is going will not stop. Some people enjoy harassing other people, so.
I think so. The harassment is horrible, and I agree. And it's not fun. And having security around you is not fun. But I like to think that the vast majority of people just don't know and are insecure and are pulled into rabbit holes because of what they read. I think that's the audience that really we should be targeting, because they, you know, if you are put next to someone. As a scientist, you say, I think this, I think that, and combined, my conclusion is such and so, and then you put next to someone who says, I know this is how it is, you know, that's a very difficult balance unless you really take people along in how that process works. So we have a program, Pints for Science. So some of our more junior staff, yeah, you may know it. So that's just in a pub talking about what fascinates you with, you know, people who are there. Yeah, and I think those kinds of initiatives are also really useful because they, you know, they make you feel what questions people have. Also, presenting for a lay audience is I think excellent way of learning, because you get the not craziest question. I mean, they are good questions, but very difficult to answer, and they put the finger on the sore spot of what you don't know. But it's also fine to then say that. So I think those kinds of things are important training to learn how to do that.
Okay. Good afternoon to everyone. I'm Francine Sado, an early career researcher coming from Cameroon. Professor Marion and Professor Edward, you are two models of success for me, and I really appreciate your presence here, and I'm really grateful for the occasion to listen from you. What I have as a question is mainly based on the problematic you highlighted. So there is a lot of research questions that came out of your presentation, both of you. And I wanted to know, as an early career researcher that is trying to find her way, I wanted to know the way of be successful in developing grant writing proposal and submitting. What are the key points to obtain some fundings when you are writing grants? Because we know that in research, there is a lot of submission that you do, and unfortunately, they are unsuccessful. So I really want to know, we have the idea, we have many research questions that we have to address, but we don't have the money for that. So I'm asking about how to become successful in research in terms of grant writing. Thank you.
Yeah, so that's an important one too, right? So I think I know that Tulio does that, I do that, you may also do that. We bring in people that are really good at that. So what they are is people actually sitting in between the scientists and the funding agency, so they know the language of the funding agency. They listen to the scientists and say, okay, if that's what you want to get funded, you have to, these are buzzwords that you have to mention. Or they also read. So we think we have a perfectly clear outline. I also have them read my outlines, and I've secured many grants in my life, but still every time I learn, because they see flaws in my logic, because they say, well, it's not my field. I know how to read a proposal. This doesn't make sense to me, or I miss how this links to what is in the call text. Can you explain that? So it's really training. It's learning by doing. And to me, that's why I like to do lectures for, let's say, lay audiences, because that helps write down things that are clear for people coming from different fields. Because I think so there are some funding schemes that are really evaluated by experts in the field, but that's not always the case. So if you think, for instance, in our European Union proposals, rarely there will be a virologist in the panel. So you have to learn to explain what you want to a broader, even expert audience. So it's things like that. It's understanding how the funder works, what they really have in, what are the buzzwords, and understanding what your own, is my idea really clear? Is it competitive? Has it been done before? It's that kind of stuff, but it's really learning by doing, I think. And finding people that can coach you with that, because that's, it's trainable skills and it's hard work. But it's fun when it succeeds.
Thank you. And I think that I can answer for Eddie, because I took his office when he left, and all this story of his legendary locking his office and in 6 hours writing a paper or grant. He was quite famous for that in Oxford. Yeah, but on the kind of one serious note and another very happy note, we're gonna close now. We're gonna have lovely food, no shellfish, otherwise we would, I hope not, yeah, we would get some norovirus, yeah, and we're gonna have something to eat and drink, yeah, and just to really, there was a reason why I really asked Eddie and Marion to be here, because they were extremely harassed in science. I was also harassed, yeah. And I think that we have to stick together. We have to highlight that we are here to really help to identify, control epidemics and pandemics, and that's not gonna stop, yeah. And as Eddie and Marion say, that's something that maybe we can, that I will talk after, maybe with VEME, maybe we can write opinion paper that we have to stop harassing scientists, and that even under harassing, we will continue doing, yeah, because I continue being quite vocal. I get in shit sometimes, but that's normal. Yeah, yeah, and that will continue, because our really job there, it is really to help to control epidemics, detect pathogens, and that. So that's something that we may even take as a collective, yeah. But as we think about that, maybe you can have great ideas how to do that, especially with some of the Stellenbosch wine outside. But otherwise, thank you so much, Eddie. Thank you so much, Marion, to be here. I must say that you guys are our heroes, or at least mine ones. Yeah. Yeah. I know that Eddie has to give another lecture for Berkeley, yeah, in a few minutes, yeah. So, and he's nervous there. But Eddie, I think that they want a photo of you and Marion, yeah, and me. Oh goodness, yeah.